Hey guys, good evening. Just kind of going over the attendance real quick. Looks like everybody's got their first and last names in, which is great. That's how we are able to assign attendance. So that's good news there. We may still have a couple people coming in, it looks like, but the attendance looks good with first and last names, so that's great. Okay. All right. So last night we kind of started off on we kind of did kind of took a pulse on how we thought the exam one went, and I kind of want to start off and get some feedback on lecture exam one. I know we hit some high topics from the beginning of the semester until now, and I just kind of want to get an idea of how we thought exam one went. So chat me in something in the chat, and we'll kind of address things as we go until we start some new material. All right. Thank you, Jam. Okay. Shani, you thought it was fine? Okay, good. That's good. So you thought, what was your, if you don't mind, can you expand on that? Like, what did you remember seeing on exam one? Did we have like a lot of chapter two, a lot of electrons, a lot of macromolecules? We talked about membrane transport. That was a big topic. Okay. Give me some ideas, Jan. Yeah, you said you maybe some soft spots from certain concepts. Do you mind just kind of telling me what you thought was a bit difficult? Maybe it was chapter one that we might have overlooked some easy material, or history of science or something like that. Um, Tell me a little bit more if you don't mind. And then if everybody seems to be okay, then we'll just kind of move on to some topics we've been talking about before, and then maybe this will be a short spotlight. I'm not sure. Yeah, osmosis. The thing about osmosis is it's also thank you, Elizabeth, for mentioning that. So osmosis, when we try to teach diffusion, we're teaching solute going from high concentration to low concentration, right? Solute concentration is from uh, areas of high concentration to areas of low concentration. So that'd be like sodium going from the outside and trying to move into the inside of the cell during, let's say, like an action potential. The thing about osmosis is we're really not worrying about what's going on with solute. We're really concerned about what's going on with water. When we talk about osmosis, we're talking about water. So if you were to put like a big salt brick in the middle of a puddle, you may notice that water gets absorbed into the salt brick, because water goes from areas of low solute concentration to areas of high solute concentration. Osmosis really explains why we have high blood pressure, if you want to think of it from that standpoint. If you think about it, when we go and we eat a bunch of fast food, or where it's all like sodium central, and we have too much sodium from, let's say, eating a bunch of country ham or something like that, our bodies will retain water, and it retains that basically through osmosis, because the salt will retain the water, and it'll remain in our circulation and lymphatics and other places where we store water, and that will that will keep our blood pressure elevated to an extent where sometimes it can be unsafe. That's one way of trying to remember osmosis. We're really just focusing on water and how it is attracted to areas of high solute concentration. It's kind of interesting when you put it in that perspective. Uh, any other points of interest or emphasis that we want to talk about from exam one? I think this week also is lab one exam, if I'm not mistaken. Some folks are doing their lab one exam. We can definitely talk about that tonight if necessary. So, Jan, is that maybe one of your soft spots was the plasma membrane and the different components of the cells? So, I think you're talking about like a phospholipid. That was the membrane, right? Where we have it's kind of like an aliphatic molecule. You might have remembered that term from chapter two. And what an aliphatic molecule, it's not amphiphatic or aliphatic. I think it's amphiphatic. Anyways, the point I'm trying to make is that one side is water loving and the other side is water hating. You know, polar, which is water loving, and then non-polar. So you have a polar phospholipid head that's pointed on the outside of the cell that's going to be facing the water. And then you have these lipid-like tails that come into the middle, and they're non-polar because they're made of fats essentially and oils, and they are non-polar or water hating. Excuse me. Pardon me. That came out of nowhere. It's only Tuesday. Um, so then we have cholesterol, obviously that we didn't really talk about much. Um, but that plays a role in the membranes, the fluidity of the membranes. And then the different components of the cells is really just organelles, right? Mitochondria, Golgi apparatus. I think sometimes students overlook that stuff because it's high school, and they may think they understand it, but then, you know, they'll leave it up to these professors or instructors to, come up with a very clever question on taking an easy concept and making it harder. So this class will definitely uh, press the limits on some easy overlooked material, normally to get their curve, in my opinion. I was a student once too. All right. So, anything else, and before we move on to some chapter 3, 4, and 5? Yeah, of course, Shauna. Let me read this. It says I have trouble with figuring out ways to remember everything because it's a lot. It is a lot of info. This class is a lot of info, and the way we can digest a lot of info is literally spending time with it. Okay, spending time with it, looking over this material every day, if not 6 days a week. If it's an hour, if it's 4 hours or 3 hours, however long, we need to spend time with the material. The more time you spend with the material, the more likelihood it's going to stick, and it's really going to stick if you start rewriting your notes. If you rewrite your notes in your own words when you're trying to explain concepts, the purpose of that is really to help put your notes in your own words. Okay, like if you want to try to explain the different steps of mitosis, you should rewrite mitosis the way you understand it in your own words and writing it out. And that's most important. That is how I think you can, bite off little bits and pieces every day to try to understand it, and then really applying it to real world facts, right? Mitosis can be applied to, what happens when mitosis goes wrong. We can kind of draw that into cancer and cell cycle. When we talked about osmosis, we can draw that into high blood pressure, right? Normally, you may need me to try to connect the dots there, but sometimes the book or the notes can do that for you. I would try to steer away from flashcards. A lot of students think flashcards are going to get them where they need to be. No, they won't. This class is not based off flashcards. It's not going to ask you what is the energy currency of the cell, or where is the energy currency of the cell made. And it may, I guess, that's like a softball toss. The answer would be mitochondria. What it's going to ask you is what happens when uh, the energy currency of the cell is low. What does the cell end up doing? It's going to try to, you're going to have to learn what the energy currency is, where it come from, and then what happens when it changes. All right. We're taking what we know and we're applying it to a new setting or a new environment. That's how you learn to think critically. And you're going to need that whether you're in nursing, surgical tech, hygiene, anything that has to do with pathophysiology where the world's not going to be perfect. All right. And things do change. So I would try to steer away from, if you want to learn flashcards, you can learn flashcards for, let's say, if you're really talking about anatomy, Shauna, that'd be like anatomical position. Like anatomical position would be, interior, posterior, inferior, um, lateral, medial. Those are your flashcard concepts because that's just something you're just going to have to know very quickly if you're trying to describe something. But if you're really talking about some pathophysiology, heart disease, elevated blood pressure, cancer, then we're really getting into the concepts where you want to rewrite your notes. So I think spending time with the material and then rewriting your notes are critical. And I would start doing that now because it may be hard, your hands may cramp up, but then by the end of the semester, you're going to be a pro at it. And then you're really going to want to do that for Bio 139. So yeah, I would try to get into that. And then honestly, you have to realize once you rewrite your notes, you really just made your study guide. Okay, and then you can refine your study guide the more times you rewrite your notes. And I'm not saying you rewrite them all, just rewrite the soft spots, right? If Jan thinks osmosis is a little bit soft and she doesn't quite understand it because she can't explain it to herself, then I would go back to my notes and rewrite some of the concepts on osmosis until you get it, and then cross that off your list and see what you're strong in and see what you're not. I mean, I wouldn't get hung up on like little minute details, but the big concepts of osmosis and cell organelles and stuff like that, definitely be able to piece that together, or the cell membrane. So I hope that answers your question there. Let me know if we gotta revisit that and critique it a little bit. Any other new stuff? Do we want to go into new stuff? Kind of a quiet crowd tonight. Looks like we only got a few folks involved in the chat, which is fine. We'll probably end early tonight. Anything we want to talk about with chapter 3 or 4 or 5? Something some folks even skipped over chapter 4. This would be like transcription, translation, and DNA replication. We had some big topics last night about translation, but hold on, before we get to that, yeah, it sure does. So yeah, Jan, so with hypertonic and hypotonic and isotonic, that's like lab exam stuff, right? That's lab 3, if I'm not mistaken. So let's go over that. So when we're really talking about, let's use the example of a red blood cell, right? That's what the book usually likes to use. And what happens is that when we have the flow of water moving from an inside compartment to an outside compartment, or vice versa, that's kind of, you can think of it as like having a lot of salt in one place, which is going to pull the water one way or the other, right? So, and sometimes when, let's say we're out in the desert and we have sweated out tremendously without refueling ourselves with water. So when we sweat, we're sweating out some salt, but more importantly, we're sweating out water, right? So when we sweat out water and we're not replacing water back, we're really creating what's called a hypertonic state, right? Where more water is sweated out than salt. and there's a higher concentration of salt left in our bodies. And what happens is that there is about 5 to 10% of water inside cells. And that water begins to get pulled out of the red blood cell or any other cell for that matter because there's so much salt outside because we sweated out all the water. Eventually, the body will recorrect or correct itself after a day or two, if you don't die of dehydration. But at this point, it has to get water somewhere, so it starts robbing water from inside the cells. And when you pull that water out of the cells, you're really creating what's called a hypertonic state, where you see these what's called creatinine red cells. If somebody were to have their blood drawn, that's usually older. Typically, more elderly people are in a dehydrated state. They never drink enough water, and you can look at their cells on a peripheral smear underneath the microscope, and you'll see. You could Google this if you want. They're called creatinine reds or creatinine red cells, and think of them as like little shriveled up peas. Rough, like green, like I don't know, peas that you eat, green peas or whatever they're called. I don't know, chickpeas. But the point I'm trying to make is they're like a little rough around their membrane. They're not smooth. They're kind of like indented in. And that is because they are pretty much been underneath a hypertonic state because somebody may be dehydrated for a long period of time. And so the body will rob that water from the red cells. And that's a hypertonic state. So opposite is hypotonic. So it's hypo means lesser. And basically what happens here is when we're just jugging gallons of water. We've taken on so much water that there's so much water on the extracellular fluid of the cells, outside the cell, that it has to go somewhere. So it begins to get pulled into the red blood cells, and you see these big puffy red blood cells if you were to draw somebody's blood. Sometimes it could be dangerous. I think water toxicity or something like that. I think that is over a long period of time, like days on end. Because basically, if you chug a bunch of water, what's really going to happen is you're either going to vomit it up or you're going to urinate it out. It's going to stay in your stomach until your stomach will, process it, and then you're just going to urinate it out. But at some point, if you do this continuously, you'll kind of create what's called a hypotonic state, where all the water rushes into the cells just to store it, just to put it somewhere, and your red blood cells get much larger because they've taken on so much water. And then isotonic is not too cold, not too hot, right in the middle, where we really have this equilibrium of water coming out of the cell and water coming into the cell. So that does kind of explain osmosis. I hope that clears things up for you. Yeah, great. Make sure I turn my speaker on in case somebody says something. My bad. All right, so let's go over and feel free, if we still want to talk about old stuff, just pitch it in the chat and we can go over it. I'm just going to touch on a few topics tonight, and if we think we're okay, then we'll go ahead and call it a night. We talked last night about DNA replication and what happens when we have transcription and translation. I think everybody seemed okay with mitosis. We can go over mitosis if you'd like, but I'd like to kind of everybody seemed okay with that last week, and I don't have much direction tonight. So we will touch on transcription and translation. So when we have DNA replication, what we're really doing is that we're creating a new DNA strand. And the purpose of that is to actually produce an RNA strand, an mRNA strand, messenger RNA. And what happens with this messenger RNA is that we're really replacing the thymines with uracils. That's how we know it's an mRNA strand, is when you don't see any thymines. You see all of uracils. And they're typically single-stranded, and they're not very stable. That's why DNA is more stable. That's why we store all our genetic material in DNA is because DNA is much more stable than mRNA. RNA is where a lot of vaccines are made out of. They'll make viral RNA. I think maybe some of the COVID vaccines are based off mRNA or messenger RNA of the COVID virus. And it's just little bits and pieces. And what that does is that it will kind of get broken down very quickly, but it'll hang around just enough for the immune system to get a hold of it and crank out antibodies against it. And that's kind of how we've created the vaccine. So what we need to kind of go over is that when we have transcription, that still takes place in the nucleus, right? We're still in the nucleus at this point. We bring in RNA polymerase, and we're reading the DNA strand to create this mRNA strand. So the RNA polymerase will read the DNA, take the thymines and replace them with uracils, and then we have this long coded uh, wording or whatever of mRNA. Now, if your book talks about pre-mRNA versus mRNA, does anybody know the difference between pre-mRNA and mRNA? Chat me something if you think you got that. What's the difference between pre-mRNA and mRNA? Did we talk about introns and exons? Yeah. Pre-mRNA is the starting mRNA. Yeah, that's right, Jan. And there's another part that we have to do, and it's called introns and exons, right? Um, introns are really just garbage code. And when we kick out the introns, splice them out, or take a spliceosome, or we cut them out, what we're really doing is that we're cutting out the introns and we're going to keep all the exons together, because that's what we really want to do when we're going to go in translation, is we want to code the exons. We do not want to code for introns, so we kick them out. And as soon as we have all the exons in the mRNA, there's two other things we have to put on it. And on the 5' end is called a GCAP, or guanosine cap, right? A bunch of guanosine nucleotides on the 5' end of the mRNA. And then on the 3' end, we have what's called a poly-A tail, right? A bunch of adenosines connected together. And basically what this is, it didn't do that last night. I don't know why I did it this time, but thank you for catching that, Jan. So I think you probably didn't hear me talk about GCAP or poly-A tail. So the GCAP really is a bunch of guanosines connected together, right? That is, we have all our exons connected, and then we have a guanosine cap on the 5' end, and then we have a adenosine on the 3' end. So we have a bunch of poly-A's, a poly-A tail, otherwise known as a bunch of adenosines. And then we have a GCAP, which is a couple guanosine nucleotides on the 5' end. And what this really says is that it will tell the nuclear envelope, and now this mature mRNA is ready to go into the cytoplasm and begin translation, right? And then once it makes its way into the cytoplasm, we're at the point where we can start bringing in tRNA, transfer RNA. And does anybody know the difference between a charged tRNA and a regular tRNA? Let's see if we know that. So the difference between a charged tRNA and a regular tRNA is that the charged tRNA actually has the amino acid on it. And what happens is that we have this reading frame. And a reading frame, and this was like I think somebody answered this. It was like a question on an exam or something like that. Is how many codons do we have if we have, or how many amino acids will be added if we have 99 codons? It's classic example of a translation question. So this reading frame that we have, what that is that it is how the tRNA. Yeah, that's right. There's different kinds, Elizabeth. So feel free to kind of help guide this conversation if you want. So we had mRNA with transcription, but now we're really talking about tRNA, messenger RNA for transcription, and then transfer RNA or tRNA for translation. So we have this mRNA that's made its way to the cytoplasm, and then the tRNA will read the mRNA in 3 nucleotides at a time. It's called a codon. All right. So, and then that will kind of establish the reading frame. Think of it as think of a reading frame the way we read letters in a book or words in a book. We group these letters together that make up words. Okay. Well, these tRNAs will read 3 nucleotides together to make up a codon, and that's what makes sense to them. Okay. So it sets the reading frame when it reads one of these nucleotides and turns it into a codon. That sets the reading frame. This really distinguishes whether we're going to have some kind of pathophysiological event like cystic fibrosis or sickle cell anemia. Both of those are serious diseases caused by what's called a point mutation. And a point mutation, all that really is a reading frame shift down one nucleotide that throws the whole reading frame off, that can code for a completely different protein and potentially causing serious side effects. In cystic fibrosis' case, death. Eventually, in our lifetime, that will be corrected. We're close to that, fixing that kind of technology. Or we're close to finding the technology to fix these point mutations in certain congenital diseases like CF. So what happens is that along the mRNA, we have 3 sites. And I remembered this as EPA, like the Environmental Protection Agency, EPA. But what they really stand for from going left to right is exit site, processing site, and accepting site, right? EPA. And when a tRNA comes in to read the mRNA, it's going to look for something called AUG, adenosine, uracil, guanosine together to make up that codon. That just so happens to be the start codon. That when it sees that code, AUG, it knows that it's going to put the first amino acid right there on that mRNA strand. And that's going to set the reading frame. Excuse me. And it just so happens that the only amino acid that is coded on there is methionine. So when a tRNA that's charged with methionine reads AUG on the mRNA, then that starts the protein. Every protein is going to begin on the 5' end of a methionine. And then it moves to the P site, right? And that's where it kind of starts. It gets accepted in the A site, moves to the processing site, the P site. It deposits its amino acid right there on the P site, and then it gets kicked off on the exit site. So now we have a charged tRNA dropping off its amino acid, and now it's just a tRNA. Now the next one comes in. It reads the next 3 nucleotides or the next codon. Could be like, yeah, it does. Yeah, it does. You're right, Jan. I forgot that small. That's kind of a big point to make. Not only does it take place in the cytoplasm, but the ribosomes kind of help hold the mRNA in place and will help kind of hold the new protein as it grows. So we have the large subunit and then the small subunit. And this large subunit will kind of inch forward, and then the small subunit on top will kind of go with it. And then it goes like this. Like that. Yeah, very good point there. I forgot to mention that. Um, so then we continue on. Each kind of tRNA comes in and see if it binds, see if it recognizes the next set of nucleotides, and then out, and then comes in and then out. Before you know it, we have probably a couple hundred amino acids attached to the P site. And then all of a sudden we reach a codon that says UAA or UGA, and there's another one. I can't ever forget. UGA is University of Georgia, and I can always remember that. That's a stop codon. UAA is a stop codon, and then there's a third one. And when all of a sudden the tRNAs read that brings in a whole other enzyme and cuts it, and there you have your brand new protein. And when you have your brand new protein, then it begins to fold up into its actual activated structure, right? I mean, and what that really happens is that we put a bunch of non-polar amino acids in the middle, and we have a bunch of polar amino acids on the outside. Polar means water loving, non-polar means water hating. So there's not going to be any water in the middle, kind of like there is a phospholipid membrane, and all the water is going to be on the outside. So all the water loving amino acids are going to fold to the outside, and the water hating or non-polar amino acids are going to fold to the inside. And then it's going to carry out its function. It could go to the Golgi apparatus, get packaged up and sent somewhere way far away, like a hormone. It can turn into an enzyme, which is a protein that carries out a reaction. You know, we have those everywhere. Polymerase is just an enzyme that's been made somewhere to carry out DNA or RNA replication. So it's kind of infinite what these proteins can do in the body. What really happens is when we have a point mutation or we have a reading frame shift is when we start making bad proteins. That, let's say, all of a sudden a bad protein never binds or folds correctly and then gets deposited in your brain, and then 80 years later you have Alzheimer's. Okay. Or not exactly trisomy 21. Does anybody know what trisomy 21 is? Just curious. Try not to look it up. All right. Well, thank you. Thank you, Jan. So, yeah, Ivana. Yeah, you're right. It's Down syndrome. Trisomy 21. Basically, what that is it's an extra, a tri. So it's an extra chromosome 21. So we have 23 chromosomes, right? Well, the 21st chromosome, so people with Down syndrome actually has a third chromosome of 21. And that just sometimes means you have an excess protein that can cause the features of a Down syndrome person. You know, I cannot think of, we've seen the facial features of a Down syndrome person. Something has to do with the palm of their hands. I believe there's a crease going between the palms of their hands that, and also there's a connection between Down syndrome folks with the high probability of them developing Alzheimer's at an early age, as well as some heart defects too. And that's one of the things when you have an extra chromosome 21 in these folks. So it's kind of very interesting. It all gets kind of tied in why we care about transcription and translation. And it can be confusing, but I think if you spend more time with it and you kind of walk yourself through it, maybe even draw a few diagrams out, then it will begin to start making sense for you. And certainly after you attend some spotlights, maybe ask me some questions or Dr. McCain some questions, then it'll make more sense. Do we have any other topics that we want to discuss tonight? Those were kind of the ones I wanted to hit on real quick. But we have a little bit of time. Well, we have quite a bit of time left. But we've kind of been quiet, which is all right. We may be okay tonight. All right, guys. Well, we'll turn it loose tonight. I think we're good to go. If anything does pop in your head and through your studying, and you want to review it, feel free to join the next spotlight session. I'm assuming we're okay on lab exams. I know we talked about osmosis, but it sounds like we're okay on lab, which is great. If anything does pop up, let me know when the next spotlight, and we can cover this. If you want this posted on YouTube because you want to watch it again, just let me know, and I'll throw it up on the YouTube channel. A lot of students tend to watch some of those when they're really good. So don't forget about that, and we will just catch you guys next time. Enjoy the rest of your week. I think it's going to be rainy tomorrow.